What I did say, I'd talk about cancer and more on the research, but I just found it was too much to try and do everything. So I am going to focus this time a lot more on cancer. There will be aspects of research in the connection of cancer, but I'll go through another stage and do much more about focusing on the research angle a bit more. So just to kind of go back to make sure we're all on the same page, we talked about this single cell making a whole human body, and that single cell has the code. It's a linear code, and it's in the form of DNA, which has 4 building blocks. They're arranged, these building blocks are arranged like beads on a string, and 2 of these beads come together to give a ladder-like structure, which can become twisted or forming a helix. So you'll end up with your double-stranded, because you've got the 2 strands, DNA helical molecule. That then, from that, you can get a subcode. So this has got all the information to make all the cells that we need, all the information to make the whole human body. From that, you can make a subcode, which is just the code for one worker molecule or protein. Still got 4 building blocks, but slightly different to DNA. And then we've got the protein molecule, which are the molecules that do the tasks in cell. They can be built like the brick walls, if you like. And these have 20 building blocks called amino acids. They're also assembled like beads on a string, but because they can all interact with each other, that gives you a 3-dimensional structure, and the precise shape of that determines exactly what this protein can do, and how it's going to be able to do that. And the critical thing then is that all these molecules can get damaged, but what ATM does is help to maintain the integrity of our DNA. So I just want to make sure you go through this nomenclature here. I keep saying your DNA is a long linear molecule. In fact, it's divided into 23 pairs of chromosomes, but each one of those kind of looks a bit like a ladder like that. And the information, so remember, that's got the information to make all the proteins that we're ever going to make in the body. And the information to make any single protein is called a gene. So we have an ATM gene, which is on chromosome 11. For AOA1, and remember, AOA1 is really the name of the disorder, and the gene is called aprotaxin. That's on chromosome 9. So we have the aprotaxin gene. And then we have an ATM messenger RNA or aprotaxin messenger RNA, and we're making the ATM or the aprotaxin protein. Sometimes it's called the AOA1 protein, but really it's aprotaxin. And just to avoid that confusion, ATM is the name of the protein, even in a human being who's making ATM, it just happens to be called ATM mutated. Okay, now what I want to focus on now is what a mutation is. What do we mean by a mutation? And it's a change in the sequence of these building blocks. And that can make, sometimes it's not of any impact because not all of this building block is making messenger RNA, but sometimes it can cause a change in the messenger RNA sequence, and hence it can cause change in one of these amino acids that are put into the protein. And that can be lethal if it completely inactivates the protein that's essential, or it could just change the function of the worker molecule. And all that comes from this change in the sequence of these building blocks. Now, I want also to be talking about accurate repair versus misrepair. We've focused on a double-strand break because that's the lesion that is activated and that ATM is dealing with, helping with. If unrepaired, a double-strand break can be lethal. And here, so this is your DNA sequence, much like this ladder with some of these building blocks. And here I've put in a double-strand break. By that, we mean a break in both of these strands. Sometimes they can be damaged to just one strand. That's a single strand break. Sometimes it can be to these building blocks, and there's a whole different array of damages that you can get. But we're going to talk for the moment about a double strand break. Sometimes that break can be repaired completely accurately and you get back to what you started with. Sometimes it's not repaired at all, although there are multiple pathways. So normally something does happen to these things, but if it's not repaired at all, it's probably going to be lethal. But sometimes the ends, various things can happen to this. The ends can fray a little bit, and you can, there are worker molecules that actually will digest these ends of DNA. And so what can happen is that you get your rejoining, but you lose some of this information. And in this instance here, we've lost this GC, and that's called a misrepair event. And that misrepair event will give rise to mutations, or it can be lethal. In this instance, the correct ends are rejoined. But another very common event, particularly important for cancer, is where the wrong ends are rejoined. Now, remember, your DNA is organized in chromosomes. So this has got all those building blocks, and it's all kind of coiled up to make it fit into a fairly tight structure. So it's chromosome 4 is a big chromosome, this is a small chromosome. And what's happened here is that the wrong ends have got rejoined, and we've got something called a translocation. And this is very important for cancer, where the wrong ends have become rejoined. And I just put over this, showed you this slide because it's beautiful technology where all these different chromosomes, remember we've got 23 pairs, can be, it's called painting often, you can manipulate the cells so they'll come out in different color. And what's happened here is that you may see this chromosome's got a little bit of light purple on it, and that one's missing, that light purple. In this example, we've got a little bit of blue there, though it's not so obvious where that blue has actually come from. And these translocation events, changes, very often they're lethal, but particularly if you lose genetic material. But in these examples, they can be viable, and they're very important events that happen during cancer. So the loss of ATM leads to this increase in translocations, and in general, so double-strand breaks can be lethal, but they can lead to mutations, being the change of sequence of the building blocks. Okay, so up to now, mainly, I focused on harmful mutations that in the ATM gene, mainly, that are disease-causing mutations. They're causing Ataxia-Telangiectasia. Some mutations may be lethal, as I've said, but some are actually beneficial. And evolution represents the selection of beneficial mutations. And you can think of that in terms of the coronavirus, which I so often give the examples of, where you may have heard of the Kent variant, where there's a mutation that allows the coronavirus to infect people more avidly, and that's beneficial for the virus, at least, not for us. And during selection, so during evolution, the selection allows the outgrowth of beneficial mutations. Now, cancer cells. Cancer cells are our own cells that have ceased to obey effectively the rules of society. They gained a state of uncontrolled proliferation. And so our normal cells know when they should be dividing and when not to divide. So the cell at the end of my finger knows it should stop dividing. My hair cells continue to divide, though I wish they wouldn't until the hairdresser's open. But whatever, your cells know when and when not to divide. Now, to achieve this state of uncontrolled proliferation, cancer cells have to acquire mutations. And it's generally thought that you need a certain number of mutations before you get a cancer cell. And I think it probably depends on the tissue. The example here is showing 4, but sometimes it might be 7 or 8 for a human cell. So basically, you've got a normal cell, it's got a few mutations, but you reach this stage where it's got here 4 mutations and it's got uncontrolled proliferation. It can now proliferate, and that's what cancer cell needs. Even once you've got to this state of uncontrolled proliferation, a cancer cell needs to continue to mutate to adapt to all its changing environment. So it may have started off life as a breast cancer, but now, or breast cancer cell, now it wants to go and invade your liver or something. So it's got to constantly change and adapt. And you can think of it like evolution fast track. And evolution involves the selection of beneficial mutations, and that's what's happening constantly in cancers. So cancer cells need to mutate avidly. You can almost say they're addicted to mutagenesis. So to enhance mutations, cancer cells do one of several can do several things. They often can increase their levels of DNA damaging. Remember the reactive oxygen species, or little byproducts of our own metabolism that form DNA damage. So sometimes they'll increase the levels of oxidative, of ROS damage. Sometimes they'll change how they copy their DNA. So you may remember I told you that when we replicate or copy our DNA, make one copy from another, errors can arise in that process. But most importantly, in this context, often cancers will downregulate or change their damage response processes, and they're the processes that restrict mutations arising. And that, of course, is the role of ATM. It's a damage response protein. So having no or diminished ATM is a step towards allowing cancers to arise. So just this really is just to remind you how ATM is doing that. Remember, I told you that it can be more like a policeman than the doctor. And what it does is adds this little, what we call phosphate group. It phosphorylates other proteins, and that can change how they fold and shape and change their function. So it sends signals to several different machineries. When there's a double-strand break present, it says, stop doing what you're doing, or sometimes it's do things you weren't doing, but stop the accident getting any worse. And what that does is increase survival and diminish mutations arising. The other important thing in the context of cancer particularly is that ATM can promote death of very badly damaged cells. This is a process called apoptosis, and this effectively helps to remove cancer cells. Though from a cancer's perspective, what ATM is doing is diminishing the possibility of mutations, and it's removing damaged cells that can become cancer cells. And I thought I'd give you this example to just highlight how ATM plays into this whole game. And P53, and this is a protein, and the P stands for protein, it's just the name of a particular protein, is a very common protein that's mutated in cancers. And it's something like 50% of all cancers in all tissues have mutations in P53. And what P53 does is it helps to promote that cell death pathway. And it also stops you progressing and doing things when you've got a double-strand break present. So you may think, oh, isn't that what I said ATM does? And it is. And it's tagged, so it's one of the proteins that is tagged or phosphorylated by ATM. So it's controlled by ATM. And what happens with P53, and P53 being called the guardian of the genome simply because it's so important from a cancer perspective. So when we've got damage, P53 levels start to rise, and that can be controlled by ATM. And that can sometimes stop cells dividing, so you stop the damage getting any worse, you allow it more time to repair, and that also promotes this cell death pathway. Now, from this, you might say, well, why isn't ATM as why isn't ATM mutated also in 50% of cancers, if this is? And the reason is because there is some redundancy. There are some other proteins that can also regulate ATM. And ATR, which is called Ataxia-Telangiectasia related, I mentioned it once before, is another protein that also regulates P53. So P53 is a bit worse in terms of what it's doing for cancer, a lot worse than ATM. But nevertheless, it shows you how ATM is regulating it. So because of all this, ATM is called a tumor suppressor gene. What that means is it's preventing mutations and helps preventing the development and progression of cancer cells. And many proteins that repair DNA damage are tumor suppressor genes, i.e. they restrict mutations arising. And for ATM, it actually seems to help more to increase your survival than to create mutagenesis. So ATM is not such a strong tumor suppressor gene. It's really important for surviving. So cancer cells, as I say, often change these tumor suppressor genes. And that's true. Some cancers change or diminish the levels of ATM. Chronic lymphoid leukemia is one such cancer, and breast that's been reported ATM changes in breast cancer have been observed as well. So losing ATM is one of the steps enhancing cancers forming and stopping them progressing. So if the AOA1 family are still with me, this is where I just thought I would take time out and explain to you about AOA1. So for the others, if you don't want to listen to this, you can switch off for a few minutes and come back in later and just give your mind a rest. So what I've talked about is a double-strand break. So this is a double-strand break. And, well, let's just go back. So AOA1 is the disorder. Aprotaxin is the gene that's defective. It's much smaller than ATM. It only has 7 exons and it's on chromosome 9. But although it's smaller, when we talked about trying to use vectors, trying to put ATM back, or ATM back, all of that type of procedure will be relevant for AOA1 as well, and somewhat easier for AOA1 because it's smaller. Now, what was important to remember that AOA1 acts on both double-strand breaks, but also on single-strand breaks. So I've shown it here with a double-strand break, but the same thing will be happening on a single-strand break. So there are pathways that rejoin these, either double-strand break or single-strand break. They're quite complex pathways. They do multiple things, but the final step is literally putting these back together again, and that's called ligation. And there are worker molecules in the cell called ligases that just join these back together again. And they're different. If it's a one-ended break, a single break, it'll be a slightly different ligase to one that rejoins the two of them. Now, critically, what happens occasionally is that this process forms an abortive intermediate. And it's a chemical reaction, really. And you get an intermediate stage of the reaction stuck there and it can't get off. That means then you can't go forward to complete the rejoining, but you can't use another pathway, you're stuck. And what aprotaxin does is remove this blockage. So, and as I said, it can do that as single-strand breaks or double-strand breaks. And I think because single-strand breaks are more common, it's probably more important for single-strand breaks. So that, I think that block generally will prevent repair. So you'll get a loss of repair rather than misrepair. So all the evidence is that Ataxia is not a strong cancer predisposition syndrome. And I talked with Keith Caldecott, who identified this in the first place, and he said there was no strong evidence for cancer in Ataxia, although I haven't done a thorough search on that. But it kind of makes sense because you've got this lesion that's causing a block, so you're not getting this misrepair. Also, obviously, abraxane is not working in that process where you're removing damaged cells. So I think it's in some ways, from a cancer perspective, not inducing cancers as ATM is. Okay, so now I'm going to actually go through this slide rather quickly because I've summarized it on the next one, and I think we've got, I'm talking for quite a long time, but if anybody wants me to go back to this and explain it in more detail, I will do. But the issue is many of the tumors in AT cells are of lymphoid origin. And what that means is they're derived from your immune cells. And so this is where I'm going to try and summarize it. During your development of the immune response, you want to recognize all the different foreign bodies, agents to which we're exposed. And you generate T and B lymphocytes that will recognize these foreign bodies. And to do that, you need to create genetic diversity. You want each T and B cell to be able to recognize a different agent, the coronavirus spike protein, or the measles virus, the measles protein, or a whole range of different proteins, which are the foreign agents to which we're exposed. And to do that, so in a sense, that is the opposite of our normal process where we want to maintain genetic stability. Here, we want to create diversity. And there's a process during the development of the immune, well, there's several processes during the development of the immune response that all help us try to achieve that. But bizarrely, it exploits and uses many of the same proteins that we use to maintain stability in our normal cells, but it does it in a clever way. So one of these processes is called the BDJ recombination. And basically the cell introduces a double-strand break and then rejoins that double-strand break such as to rejoin the wrong ends and to cause rearrangement and slight differences at the junctions. And that creates the diversity of our immune response. ATM is perfectly, or AT cells do this. If you don't make your T and B cells normally, you're called SCID, severe combined immune deficient. I mean, AT cells are not SCID. And AOA1 doesn't seem to have that immune deficiency either. And I think that's probably because although they may not do all the rejoining quite normally, most of it is happening. AOA1 is only used for a subset of those rejoining events when there's that stalled intermediate, and it's probably not enough to give you maybe a few less T or B cells, but that's really not going to matter. But for AT, the important aspect is that there's the quality control in AT is aberrant, and basically you can get incorrect rejoining of those ends, much like I showed those translocation events, and you also fail to remove inappropriate cells that can arise during that process, and that can give rise and allow the proliferation of lymphoid cancers. There's a second step called class switch recombination, which I'm not going to go into in any detail. I don't think that's significant for cancer, but AT individuals can have a reduced efficiency of doing that, and that leads to the IgM and IgA deficiencies. So now we can think about what types of cancers arise in AT and why. And AT individuals have mainly lymphoid cancers, but some solid cancers. The lymphoid cancers, I think, are because they have a shorter development time, or one reason is they have a shorter development time. And so they tend to be more common in children, even children without AT. So they're seen more in AT individuals. But then I think on top of that is this quite significant aspect of there's a role of ATM during the development of your lymphoid cells, and that particularly these T and B cells. So the absence of ATM then enhances mutations, the possibility of mutations in all cells. And because cancer is addicted to mutations, and because ATM has this tumor suppressor role, cancer cells, AT cells can have increased frequency of all cancers. And because AT individuals are living longer, we're beginning to see solid tumors now in some of the AT individuals. There seems to be some tissue specificity, particularly breast cancers, and the explanation for that, and I'll come back to that a bit later, but it's possibly because these are hormonally driven cancers, and there's some evidence that the hormones result in double-strand breaks arising, so it may increase those events happening. And then variant AT, lymphoid and solid tumors, get more solid tumors effectively, just because they're living older. Okay, so that's talking about why we're getting more cancers. I've talked now about that lymphoid specific aspect. What I'll now talk about is the situation for carriers. And first, I'm going to go through this slide that I introduced you to before, which is to make sure you understand what I mean by a carrier. So we've all got 2 copies of chromosome 11, and a carrier has one, what we call wild type copy, and 1 mutant copy. And then when you have children, 1 of you in 4 will have 2 mutant copies of ATM. 1 in 4 will have a wild type, 2 wild type copies, but the other 2 will be carriers. But remember, this is a numbers game, so this isn't necessarily going to happen if you just have your 4 children. So carriers, therefore, have a good copy and a bad copy. And mainly, the level of the protein depends on the number of genes. So a person with a gene that makes no copy, no protein, will have half the overall level of ATM protein than compared to a normal individual. Most of the cellular studies that are done would say there's enough ATM protein to do most of the things, even if you've only got half the amount. When we look at various aspects in the lab, using the cells from carriers, we can't see any difference. If you've got really sensitive assay, you can see half the amount of that tagging of the protein, but that may not have very much relevance. But of course, cancer is a rare event, and you only need it to arise in 1 cell, and you may well see the cancer. So in a sense, I think it's just a selection thing, a numbers game for why cancers, you maybe see cancers in carriers, but for most other aspects, they're normal. So how do we know there is a difference? And there's 2 types of studies. One is called epidemiology, which is really the study of populations. So you can look at carriers versus controls. And here, carriers, these were earlier studies before we could do all the sequencing we're doing now. And carriers were identified as being the parents of AT individuals. And they particularly looked at breast cancer, simply because breast cancer was a bit of a more common cancer. And Swift was the name of the scientist who drove these early studies, and he showed there was an increased risk of breast cancer with a potential link to mammography. And that is not completely unsurprising, although I think this has still not been particularly solidly consolidated. The increased breast cancer, I think, is real, but whether there's a link to mammography. The mammography is because it's like X-rays. You get double-strand breaks put in during those X-rays, and as I've told you, ATM is having a role in the response to double-strand breaks. Uses very low doses of X-rays, so you don't get many introduced. It certainly won't do any killing, but there may be a link to enhancing the prospect of cancer. The other type of studies are more recent ones, and they're called GWAS, and that stands for Genome-Wide Association Studies. And basically here, you're sequencing. You may not sequence your whole genome, but often you'll just look at a subset of the tumor suppressor gene that I told you about, and either sequence them or look to see if there's a linkage to into cancer patients versus non-cancer patients. And I should say here, you're not looking at the cancer cells, you're looking at the normal cells. So are those normal cells more likely, are mutations in them, or variants in them, more likely to be seen in cancer patients versus non-cancer patients? And what those studies have shown is that the mutations in BRCA1 and BRCA2, and these are also carriers, so they've only got one mutant copy, explain something like 20% of the familial breast cancers. So these are really quite high, giving you quite high predisposition to breast cancers if you're carrying those genes. And then there's variants of a number of genes, of which ATM is one, that can explain 2 to 5% of the familial breast cancer. And a lot of the familial breast cancer is not understood. It's probably not a single gene, it's probably multiple genes. And so ATM's contribution here is 2 to 5% of the total with these other genes. I'm not quite sure what it is for ATM alone. And it seems like, from reading about this, the estimates are that you're maybe as a carrier, you're 2 to 4 times elevated risk above the baseline for a normal individual. Any risk caused by mammography from these studies is unclear, and I think would take that to mean that it must be very small, because otherwise I think it would be detected. There's some work in the literature where they're looking at whether the risk for carriers with specific mutations, for example, variant mutations versus null mutations, are different. And there's some studies which say there may be some mutations that are particularly prone to giving you breast cancer. But and Malcolm Taylor and some other people have been involved in this, but I'm not going to include that study, partly because I don't quite know all the data. Now, the question then becomes of how should carriers be treated? And there's, as far as I can see, it's a bit mixed evidence here. There's no consistent evidence for increased risk of breast toxicity following radiotherapy. So radiotherapy is the mainstay, one of the major treatments for breast cancers. And the question would be, because we know AT individuals are very sensitive to radiotherapy, would the carriers also be? And there's no strong evidence, as far as I can see, that is the case, although I have seen some data saying that is the case. But there's some studies that show there might be increased cure using radiotherapy, and I'm going to come back to that. And I think this is something that actually needs to be substantiated and looked at, but I'll come back to the rationale for that in a minute. There's the question of whether if you've got a cancer in one breast, you get it in the other breast, and that can be the case for BRCA1 and BRCA2 carriers, but there's no evidence that that's the case for AT carriers. So the question then is, what's the recommendation for treatment? So for quite a number of BRCA1 and BRCA2 carriers opt to have breast surgery to avoid getting the tumors in the first place. There's no recommendation for that with AT carriers because your risk is too low to make it worthwhile. As it seems like there's a suggestion to have annual mammography to be checked and get that started at a slightly earlier age for those with the family history. And there is potentially a risk from that mammography, but it's very small and it's a hugely important tool. And the benefit is much more to be able to detect it early. Obviously, if you can have MRI scanning, then that's good because that's not introducing double-strand breaks. So why is it that there might be the risk of breast cancer? So this question's been asked a lot for BRCA1 and BRCA2 carriers that were predisposed to both breast and ovarian cancer. And it's not really that well understood, I don't think, but one of the possibilities is that they're hormonally driven cancers, and the hormones can, during their sort of usage and the way they function, introduce double-strand breaks. So there may be a case, and what I should say is both BRCA1 and BRCA2 are damage response proteins, and they're both functioning with to deal with double-strand breaks as well. Slightly different to the way ATM's working, but they're all dealing with these lesions, same lesions in the DNA. And BRCA1, in fact, is one of the proteins, another protein that ATM tags and alters. So we're just going to talk about this question of whether the tumors could be more radiosensitive in ATM carriers. Now, what's known for BRCA1 and BRCA2 is that, so they're carriers, you've got 2 copies of your whatever chromosome those genes are on. And in the tumors, you invariably lose the other copy. So tumors gain all these mutations, and one of the things they do is make you pretty much completely lose BRCA1 or BRCA2. Normally, they're essential for growth, but they make other changes so you can grow without it. And that's actually been very much exploited for therapy. And it's found that if you don't have BRCA1 or BRCA2, you lose a specific repair pathway. And there are drugs that make you more reliant on that repair pathway, and they're called PARP inhibitors. So your normal cells will have BRCA1 or BRCA2 because they're carriers, but the tumors don't. And by treating with these drugs where to survive, you need that pathway, you can then selectively kill those particular tumors, and that's done together with radiation as well. 9 times out of 10, the tumors change because they're addicted to mutation and overcome that, but at least it is working pretty well. So the question is, what happens with ATM? For the carriers, when you get your tumor, do you lose the other copy of ATM or inactivate it? And there's definitely some evidence that you do, but I'm not sure it always happens. But if you do, then you would expect your tumor to be radiation, to have no ATM. It should be very radiation sensitive, that the rest of your cells in your body will have one copy. It should be more or less normal for radiation, maybe a little bit sensitive. And I think this is something that needs to be looked at quite carefully, because it could be a form of treatment there that would be helpful. So just more or less to finish, I'll talk about the treatment of cancers in AT individuals. So cancer cells have been known for a long time to be sensitive to DNA damaging agents. And that was initially thought to be because they grow faster. And if you grow faster, you have less time to repair that damage and you're more sensitive. But more recently, it's been realized that it's this change in their damage response processes that they need to do to gain mutations means that some of those pathways are not working very efficiently. And so by trying to create mutations, they make themselves sensitive to certain DNA damaging agents. And so often those DNA damaging agents are given to particularly treat tumors. And that's been a mainstay of treatment for certainly lymphoid tumors. Now, of course, for AT, that's bad news because those AT cells may be sensitive to some of those treatments if double-strand breaks arise. So it's actually makes it much harder to try and treat some of the tumors in AT cells, and you have to be very careful about the agents that you give it. I think there is the opportunity here for some real work because you can try and exploit that for therapy. But it needs some very specific studies to benefit from that. And obviously radiotherapy can be an issue for AT cells, AT individuals, and it might be okay, but you need to change the dose dramatically. But that's not so beneficial for lymphoid tumors because they're circulating. So that radiotherapy isn't usually so much used for lymphoid tumors. Okay, so just to finish, I've got this fairly simple slide. DNA damage can affect your DNA, and if you could respond to that normally, you just maintain genomic stability and you try not to get too many mutations. If you don't have ATM, you can have an abnormal responses to damage, and that can, although often that is lethal, and that's bad news for some of the other features of AT, but from a cancer perspective, lethality is actually a good thing because you can't be a cancer cell if you're a dead cell. But what's important here is this genetic instability, the getting chromosome aberrations, chromosome translocations, gaining mutations, and that is a step that's leading towards cancers. Okay, so I'll stop there and